Every multicellular organism faces a massive logistical challenge, keeping trillions of individual cells continuously supplied with oxygen and glucose. To solve this problem of scale, the human body relies on the cardiovascular system, a macroscopic fluid distribution network. This distribution relies on two distinct but connected circulatory loops. As shown here, the systemic circulation delivers nutrients to the body, while the pulmonary circulation routes blood through the lungs to acquire oxygen. Separating these two loops ensures that oxygenated, nutrient-dense blood is not diluted by returning waste-rich blood before reaching its destination. The heart acts as the central hub, connecting respiratory exchange in the lungs to specialized systems like the liver and kidneys. Tracing the red pathways downward from the aorta shows this central line branching out into peripheral tissue beds. While this systemic highway manages the large-scale delivery, the final destination is a single target cell waiting in the interstitial fluid. Metabolic function requires the integration of this macroscopic pressure system with localized microscopic chemical gates. The driver of this delivery system is the heart, a dual-chambered mechanical pump that powers both circulatory loops. During the relaxation phase, or diastole, internal pressure drops, allowing blood to fill the atria and ventricles. Here, arrows 1, 2, and 3 track deoxygenated blood entering the heart's right side. During systole, the muscle tightens, increasing pressure to close the mitral and tricuspid valves. Arrows 9 and 10 show the left ventricle ejecting oxygenated blood through the aortic valve. This creates hydrostatic pressure, the physical force required to propel glucose-dissolved plasma through the body. Without the mechanical force of systole, chemical nutrients would remain in the chest cavity, and cellular diffusion would not occur. The liquid medium carrying these nutrients is plasma, which dissolves glucose and transports the body's blood cells. This plasma travels through vessels designed to withstand the high systolic pressure generated by the heart. This diagram compares the structural requirements of outbound versus inbound vessels. In panel A, the artery contains a thick layer of smooth muscle, the tunica media, to withstand significant outward pressure. In contrast, panel B shows a vein with a thinner structure for low-pressure return. The histological photograph in panel C shows this difference in biological reality, a thick-walled artery next to a thin, collapsed vein. These large arteries progressively branch into smaller arterioles to distribute blood across specific tissue areas. The thick arterial walls that protect against high pressure also act as an impenetrable barrier, keeping nutrients trapped inside until they reach the exchange site. At the tissue level, thick arterioles transition into capillaries, vessels with walls only a single cell thick. These capillaries are the sites where glucose leaves the plasma to enter the surrounding interstitial fluid. This illustration shows how typical capillary architecture facilitates this transfer. Porous gaps between the endothelial cells allow for the passive diffusion of water and dissolved glucose. However, the central nervous system requires far more restricted access. The brain capillary on the right lacks these standard pores. Instead, it features tight junctions and an enveloping astrocyte, forming the structure known as the blood-brain barrier. While capillary structure dictates regional access, reaching the fluid outside a target cell is only one part of the metabolic process. Because dissolved glucose is hydrophilic, it cannot cross a cell's lipid bilayer without assistance. Cells use specialized membrane proteins called glucose transporters, or GLUTs, to move the molecule across this barrier. These proteins share a common structure, a single chain that weaves back and forth through the membrane exactly 12 times. When glucose binds to the exterior of the protein, the transporter shifts its shape to release the glucose into the interior cytoplasm. This mechanism is known as facilitated diffusion. It is a passive process that relies on existing concentration gradients rather than consuming cellular ATP. These GLUTs act as passive doorways, ensuring that only targeted nutrients can enter the cell. Basal blood glucose is relatively uniform at 5 millimolar throughout the body. Yet different organs extract vastly different amounts of fuel from this same supply. This regulation is driven by the Michaelis-Menten constant, or Km, which measures a specific transporter isoform's chemical affinity for glucose. Panel A shows the kinetics for GLUT1 and GLUT3. These transporters, primary to the brain, have a Km of 1 millimolar, meaning they reach maximum transport capacity at very low concentrations. Since a Km of 1 is well below the basal 5 millimolar blood concentration, these brain transporters are always fully saturated, ensuring a constant energy supply. Skeletal muscle has different requirements. Panel B shows GLUT4, which has a Km of 5 millimolar. Because its affinity aligns with normal blood levels, GLUT4 is rate-limiting. Its presence on the membrane must be actively increased by insulin or exercise to boost uptake. Finally, the liver and pancreas must sense blood sugar spikes. Panel C shows the curve for GLUT2, which has a high Km of 15 to 20 millimolar. This low affinity ensures that the rate of glucose entry is proportional to blood glucose levels, allowing these organs to act as physiological sensors. Human metabolism relies on the integration of these two scales. Macroscopic cardiac pressure delivers the fuel across the body's distance, while microscopic kinetic affinities dictate the specific cellular rate of consumption.